Today we're going to talk about meiosis. Meiosis is a reduction division. We're going to go from an original set of chromosomes to half the number of chromosomes. In other words, if we look at a full set of human chromosomes, we see that there are 23 pairs of chromosomes, which makes up a total of 46 chromosomes for the human. Now, this is an important thing to remember. All chromosomes exist in pairs. They have two separate different chromosomes for each pair. Why is this important? Well, let's take eye color. Perhaps the gene for eye color is here on this orange slot on pair 9. In order to have the proper eye color, you have to have a gene from each chromosome in a pair. So we're going to follow pairs. Now, this is a very complex organism, the human genome. So we're going to look at an imaginary organism that has only two pairs of chromosomes, the first pair and the second pair. And all of the traits are going to be on those chromosomes. And here is our imaginary organism's chromosomes. The red and blue are pair 1, so this would be chromosome 1 and chromosome 2. The green and yellow are pair 2. This is chromosome 3 and chromosome 4. Pair 1 made up of chromosome 1, the red one, chromosome 2, the blue one, and the second pair of chromosomes made up of chromosome 3, the green one, and chromosome 4, the yellow one. Why am I calling them 1, 2, 3, 4? The first chromosome, chromosome 1, chromosome 2, chromosome 3, and chromosome 4 in two pairs. This cell is currently in its diploid condition. It has two chromosomes from each pair. Before we begin meiosis, we have to replicate each of these chromosomes. So we'll have twice as many chromosomes as we need in the cell. Now here we show the replicated chromosomes. We have two copies of chromosome 1, two copies of chromosome 2, two copies of chromosome 3, and two copies of chromosome 4. Remember, this will make up the first pair of chromosomes. This will make up the second pair of chromosomes. But we have basically two copies of each pair. Now, when they're joined here by this centromere, these are sister chromatids. All that means is two chromosomes are currently joined. So this would be prophase 1. And in prophase 1, all we're showing is that we have duplicated all of our original chromosomes, getting ready for the first part of meiosis, which is meiosis 1. Now here we see the chromosomes lined up along the diameter of the cell, but unlike mitosis, we're not pulling apart each pair of chromosomes. Rather, we're pulling apart two copies of this chromosome to this side and two copies of this chromosome to this side. So what you're going to see is two copies of chromosome 1 over here and two copies of chromosome 2 over here. Now the difference is that these both belong in pair 1. So on either side, even though we're going to have two chromosomes on either side, it's not a complete cell, because in order to be a complete cell, you have to have a copy of chromosome 1, 2, 3, and 4 in each side. These are my centrioles and spindle fibers. And here I have labeled the centromeres. We have 4 of them, 1 for each pair. Then the centrioles on either side. We have 4 spindle fibers and 4 pairs of sister chromatids. Now, just as a comparison, I want to show you a copy of mitosis metaphase. Mitosis is different from meiosis, and this is the way they line up. Whereas in meiosis, we lined up 4 chromosomes, 2 pairs of chromosomes. Here, we're lining up all 4, and they'll all be pulled apart separately into 2 identical cells. Here is anaphase 1, in which the 2 copies of chromosome 1 are pulled towards this pole, 2 copies of chromosome 2 are pulled towards this pole, 2 copies of chromosome 3, 2 copies of chromosome 4. Now here is telophase 1, and we can see our little cleavage furrow, right? And here are 2 copies of the first chromosome and 2 copies of the second chromosome, 2 copies of the 3rd chromosome, 2 copies of the 4th chromosome. So now, even though we've got 4 chromosomes that are going to be on either side, we only have 1 chromosome from each pair on either side. So we're effectively cutting in half the amount of chromosomes in the cell so that we can make a sex cell, either a sperm or an egg. Now here we're in prophase 2, and we're going to have to split these things again to get our sex cells. Now you notice we have 2 red chromosomes and 2 green chromosomes on this side, 2 blue and 2 yellow. What we should have in mitosis would be a red, a blue, a green, and a yellow. But we don't have that, so we've only got half the number of chromosomes from each pair. These will be split further when we go to metaphase 2. And here they're lined up to be pulled apart again. There's our centrioles and our spindle fibers. And these are still sister chromatids. This is chromosome 1, chromosome 2, chromosome 3, and chromosome 4. And remember, we have 2 copies of each chromosome. And these would make up pair 1. These would make up pair 2. Here is anaphase 2. And in anaphase 2, you can see that we're pulling apart the chromosomes. Now, why are they bent in this direction? That is just to signify that they're being jerked backwards rather quickly and forcefully. That's the only significance to the bent chromosome shape there. The only reason I address that is I've had some students ask about it, that it supposedly confused them because they couldn't figure out why these were bent and the other ones were straight. So they're being pulled to either side of the pole. Here we are in telophase 2, and we can see now that we have 1 copy of each red chromosome, 1 copy of a blue chromosome, greens and yellows. So on either side, we're going to have 1 copy of a chromosome from each pair. Our final result after meiosis is haploid daughter cells. 4 cells with half the number of chromosomes that the original cell had. The original cell had a red, a blue, a green, and a yellow. We duplicated that, split them apart, then split them apart again, and we ended up coming up with 1 chromosome from pair 1 in each cell, 1 chromosome from pair 2 in each cell. Hopefully this clears up the mystery of meiosis.